In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. How are you, dear students? I hope you all are fine and enjoying your health and studies together. Dear students, I welcome you in Pakistan International School Taif's virtual learning system for the session 2020. And you are studying biology, and as you know, this is class for second year boys and girls. Dear students, in today's lecture we will start our new chapter, that is chapter number 21. Chapter number 21, and that is about development and aging. And in this chapter, we will see embryonic development. So let's get started. Dear students, development and aging is in not only in human beings, but in other organisms also. But we are talking especially and specifically for human beings in this chapter. Development is a series of changes which animals and other organisms undergo in their passage for the embryonic state to maturity. So from embryonic state to maturity, all these changes are included in development from a lower to higher state of organizations. We will talk in this chapter all about the development, how this is done, how it is started. We have seen in the previous chapter reproduction also, that how this is started by the fertilization of egg and sperm. Aging is the process that is said to be senescence or aging. Senescence is also the name of aging, and its study is called gerontology. It is a gradual deterioration of functional characteristics. So this word can refer either to cellular senescence or to senescence of the whole organism. So actually that is, we can say, the negative development. Development is not only positive, but it is negative also. Positive, like changes in the body, that is called development, and negative changes after a specific time, that is called aging. So we will talk about this process from the development up to the aging process. So first step of the development of an organism, that is embryonic development. So embryonic development, also called embryogenesis, and its study is called embryology. That process by which embryo forms and develops. And embryonic development starts with the fertilization, as I've mentioned before, fertilization of egg and sperm. Together they make the diploid cell, and then diploid cell starts to undergo embryonic development process. Once fertilized, the ovum is referred to as zygote and single diploid cell. So embryonic development in human, it covers the first 8 weeks of the development. At the beginning of 9th month, embryo becomes fetus. Now we will see embryonic development. How does it started? Its first step is, or first stage actually, that is not a step, that is a complete stage in which the different processes are going to be done. That is a cleavage process or cleavage stage. Cleavage word comes from cleave. It means breaking down or distribution or breaking off any cell, we can say. So the first major phase of embryonic development. So it is the rapid succession of cell division. It creates a multicellular embryo from the zygote, and there is no growth. So if we see here, sperm enters the eggs and that becomes fertilized and it just forms a zygote. Then zygote is divided into two cells vertically, and then after 1 day, 1 to 2 days, it is divided into, it just carries 1 to 2 days. So it carries 1 to 2 days for the first cleavage process, that is vertically, and that is like happened when one cell is divided into two cells. Then after 2 to 3 days, there is another stage, like again vertical division is done, that is called um, a second cleavage, and 4 cells are formed in this case. Now, after 4 to 5 days, the next going on cleavages are occurred, and okay, 4 to 5 days. Before that, 2 to 3, and the 3 to 4 days, then horizontal cleavage is occurred, and then 4 cells are divided into 8 cells. So when this cell is divided horizontally into 8 cells, after this the cleavage process is we can say rapid, and after 4 to 5 days there are many cells, division is done, and a solid mass or solid balls of cell is formed, that is called morula. So that is the first stage of, we can say, the proper form of that cells. That is called a solid ball of the divisions of the cells. Then after 5 to 6 days, the morula is going to be differentiate into um, some layers, and that is called the single layer of cells surrounding by fluid filled cavity, that is called blastocele. Now we can see this mass of cell, that is called morula. Now that is further divided and divided into several number of cells, and then the mass of balls which we can see from outer side, that is called blastula, that is hollow ball. Hollow ball means that blastula, if we see its cross section, so that now that is all the cells just going to be just accumulated on the outer side, and inner side is empty, and that is fluid filled, this one. So that is called blastocele, means the cavity uh, of the blastula. This all is happened by the process cleavage, and we can say that after to 6 to 7 days or 8 days, we are gonna see this type of structure. Now here, the cell is going to be differentiated into different layers. So inner cell mass, which is going to be produced here, that is called embryoblast, and that produce embryo later on. Here, the mass of cells going to be appear here. So these cells are called embryoblast because that produce embryo, and the outer layer of the cells that called trophoblast. So the trophoblast cells is uh, that is helping in penetration into the uterine wall, or implantation into the uterine wall, or we can say that is going placenta is going to be formed by these trophoblast cells. Okay, just talk about a cleavage, some more points regarding that, that if the egg cells is having the amount of yolk, some are having, like in birds, there is more, we can say, they are having more yolk. And if we just see in the mammals, they are having very less yolk amount. So how do they uh, cleave? So different types of animals having different amount of yolk in their eggs. So there are the four types of egg cells, like isoleucine. Isoleucine means they are having very little yolk and that is evenly distributed. Iso means same, leucine means yolk that evenly distributed. Meso means moderate, leucine means yolk, so it is having moderate amount of yolk, and but that is concentrated at vegetal pole. Okay, what is vegetal pole? There are two types of pole, like when the horizontal division is done, so we can see that the cell is divided into two regions. In one portion, the cell is divided very rapidly and there is less yolk. That is called animal pole. And the other is where there is yolk present, like proteins and fats are present. So there is hardly distribution of the cells. So the cells are less in number, but they're larger in size. So that is called vegetal pole. So they animal pole cells divide rapidly and that forms, we can say, the embryo, and yolk cells that divide very slowly due to the presence of yolk. So that is vegetal pole. So in mesoleucine cells, the yolk is concentrated towards the vegetal pole. That's why here the division is very slow. Teloleucine, lots of yolk is present at the vegetal pole. In mesoleucine, yolk is present in animal pole as well as in vegetal pole, but more in vegetal pole. But in teloleucine cells, uh, yolk is only present at the vegetal pole because there is too much yolk, like in the birds. So central acetal cell, lots of yolk, but that is located centrally. So here, if we just talk about cleavage planes in all these, how the cleavage pattern is done. So cell division is slowed in yolk. So yolk can cause uneven cell division at the poles. There are two type of cleavage if we just talk about the amount of yolk. So one is like in holoblastic cell, that is complete division, in which where uh, occurs in species whose eggs have very little or moderate amount of yolk, such as sea urchins or frogs, like amphibians are also like present in this category. Meroblastic cells, incomplete division of eggs, and occurs in species rich in yolk, for example, reptiles and birds. So human beings also comes in holoblastic cleavage, incomplete cleavage. So you can see here in starfish, isoleucine yolk is present, so equal distribution, we can see equal distribution or equal cleavage in all the cells. But in mesoleucine cell, we can say, because that yolk is concentrated on the vegetal pole, so here the division is very slow. We can see here this side, the vegetal pole, there is less division, but in animal pole there is more division. Then there is a teloleucine egg, in which uh, just remember chicken's egg which we eat, there is a very small portion on the yolk, where the cleavage can be occurs. That is incomplete cleavage. So you can see with this, yolk is evenly distributed in sea urchin. Blastomeres are there. Blastomeres are actually the small cells which after mitosis and after cleavage, which are formed at that time. So these are all blastomeres. So here that is a complete cleavage also here in the frog, but that is mesoleucine eggs. Then we see the next step of this embryonic development, that is gastrulation. So the most characteristic event occurring during the third week of gestation is gastrulation, like in the pregnancy. That is, gastrulation. And in this gastrulation, it is the process that establishes all three germs layers. These germs layers are ectoderms, mesoderm, and endoderm. As we know that endoderm is the innermost layer, ectoderm is the outermost layer, and mesoderm is the layer between these two, ectoderm and endoderm. So that is, we can say, the first process of morphogenesis also, or which we studied in morphology, like structure formation. So morphogenesis is a process whereby individual cells undergo complex movements that generate the organ rudiments. So from these organ rudiments, the proper organs are formed. So gastrulation generates the three basic germs layers. So how do sheet of the cells or epithelia move during gastrulation, or like we can say how there are these germs layers are going to be formed. The first process is called invagination. Okay, in invagination, we have to see just first that in human embryo, gastrulation begins at the 15th day of the fertilization. So during this phase, trophoblast cells, um, or the cells which are around the outer layer, that thickens at one point to form mass of the cells, that is called inner mass cells. So after implantation, the inner mass cells grows and splits and forming two fluid filled sacs that are separated into double layer, that is called embryonic discs. So here invagination means the local inward movement of the cells. Which cells? These cells. These are called trophoblastic cells, which are around them. How they move inside the movement, by the movement and completely invagination of these cells, we can see here the structure, and then involution that is similar but more dramatic, an inward expansion of epithelial around the edge, and that is such as blastopore. Then it will form blastopore here. This is blastopore, from which these cells move inward completely, and that is formed endoderm. How this process is done, we will see here clearly so that you can understand it properly. There is gastrulation process. So it begins with the formation of primitive streak on the surface of the epiblast cells. Okay. Here is the cell, this layer of the cell that is called as trophoblastic cells, as we have seen in blastocyst. While it is moving towards the uterus for implantation, here another layer of that is formed, that cells are called embryonic disc here, which later on formed in embryo. And these cells actually move inward, and that from this side they are going to be implanted into the uterus. Now this embryonic disc, this is just divided into two layers here. These two layers, okay? And these two layers, if you can see, this is structure, not this one actually. So here this embryonic disc, inside there is a cavity, and then this embryonic disc is divided into two layers. So that layer is called as epiblast and hypoblast. The upper layer is called epiblast and the lower layer is called hypoblast. This embryonic disc is like divided into two layers, epiblast and hypoblast. Now, what will happen? When these cells are going to move inward, first of all, primitive streak is formed. This is primitive streak, and this is from where, if we just move this upward, this is embryonic layer, that is called epiblast, and this one is called hypoblast. So what will happen from here, from this epiblast, primitive streak is going to be appear, because that these cells, these are formed by like the upper layer, and that forms later on ectoderm. We'll just talk about, like these cells starts to move inward. Like a line appears here, that is called primitive streak, and that is clearly visible as a narrow groove with slight bulging regions on either side of the caudal end of embryonic disc. Of this embryonic disc, this will appear like that. You can see here the cross section of this one. So it appears in 15 to 16 day of the embryo. The cephalic end of the streak is the primitive node. This is primitive node. It means that this will form the caudal end and this will form the cephalic end. Like here the brain will be formed, and here the our lower limbs will be formed. So it consists of slightly elevated area surrounded the small primitive pit. This is called the primitive node, and inside this is called primitive pit. Now this primitive pit is used to for the invagination. So we can see here these are epiblast cells and these are hypoblast cells. So these epiblast cells, they are going to move like this and make the primitive streak, and on this like epiblast cells, the cells of ectoderm moves towards inside. You can see here the cell is going to move inward and more inward. So when they move inward, they migrate and that replace these hypoblast cells. When they replace these hypoblast cells, so this will form endoderm, this will form ectoderm, and this will form mesoderm. If you see here clearly, mechanism of gastrulation, cells of epiblast migrate towards the primitive streak. Upon arrival in the region of the streak, they become flasks shaped like this, flashes, with that they can move inward easily. This is actually epiblast or future ectoderm. Now this will move, the cells will just accumulated here and move towards inside from this primitive streak, and here is Hansen's node, or that is called primitive node. Then they move again deeply inside and replacing these cells. These are hypoblast cells, or these are called endoblast as well. So these endoblast displacing endoderm displacing endoblast cells. So these cells become just present here. They just form their place at this place, and they attach here and they form endoderm. Some cells move inward because there becomes a cavity, and this becomes mesoderm. So once the cell have invaginated, some displace hypoblast, creating the embryonic endoderm, and others come to lie between the epiblast and newly created endoderm to form a mesoderm. And the remaining is called ectoderm. Ectoderm cells are the same, but they remain up on like on the epiblast cells. So epiblast, through the process of gastrulation, is the source of the germs layers actually, and later on they will give rise to tissues and organs of the body. Here we can see another detail of this one. If we see here, the primitive streak is appeared, and these epiblast cells move inward, and they start to replace the hypoblast cells as well. This is called endoderm. Some just remains here. This is extra-embryonic mesoderm, or the here intra-embryonic mesoderm. They are having different fate of forming the organs. Then there is epiblast that will be the future ectoderm. If you see this image here, so you can see clearly the movement of the epiblast cells. And this is primitive node and primitive streak. So this is the upper view of that one, primitive streak. So from where the cells move inward and move inward deeply inside that towards the caudal end, as well as that is cranial end. Later on that will form oropharyngeal membrane, and here will the cloacal membrane. So here we can see the gastrulation properly. Like this is the trophoblast cells. These are the inner mass that is called embryonic disc. And then embryonic disc is further divided into this one that is epiblast and hypoblast. Then this will start to move inward deeply inside. And as you can see here, deeply inside, and then start to form three germs layers: endoderm, ectoderm, and mesoderm inside. I hope you just understood it well. Now we will see these fate of these three germs layers. The ectoderm that will give rise to epidermis of the skin and its derivatives like epithelial lining of the mouth, rectum, sense receptors in epidermis like cornea, lens of the eye, nervous system, and adrenal medulla, tooth enamel. So this nervous system that will be formed by neurulation, that is also from the ectoderm. Now, endoderm, epithelial lining of digestive tract, epithelial lining of respiratory, liver, pancreas, thyroid, parathyroid, all the glands are formed, and all the linings of the all systems that will be formed by endoderm. Then mesoderm, that will give rise to notochord in animals retaining it as adults. So notochord will retain in our, we can say, the backbone in the adult stage as well. Then it will give rise to skeletal muscles, skeletal system, muscular system, excretory system, reproductive system, and except germ cells which are differentiate during the cleavage, and dermis also. These are all epidermis of the skin will be formed by ectoderm, and dermis, because there is inner layer, that will be formed from the mesoderm. So these are given here also: endoderm, ectoderm, and mesoderm cells. Now the next step of that is very, very important, that is called a neurulation, the first major event in organogenesis. So give rise to brain and spinal cord, more specifically, and ectoderm folds inwards as a neural groove with neural folds. That is in the 22nd day, neural folds fuse to form neural tube, and at the anterior end and brain, and rest of at the back at the that will be formed into spinal cord. Now we'll see the process of neurulation, that is also called the first step of organogenesis, and that is very, very important. Organogenesis means the formation of organs starts from here. So first of all, we'll see this process. This is ectoderm, which is formed after gastrulation, and this is the part of mesoderm that is called notochord. Actually, notochord is the stimulant for the formation for the neurulation process. That is very, very important because it helps in the formation of that nervous system, and later on it is just changed into, transformed into the nucleus pulposus, which we have seen before in the chapters in our backbone. Now we will see the process. Number first step is a neuroectodermal tissues. Like here, the ectodermal tissues differentiated from ectoderm and thicken into the neural plate. Now these cells thicken up in the neural plate, as we see this one, and the neural plate border separates the ectoderm from the neural plate. Now we can see this is the mesoderm. This is actually the cells which are just seen in this shape. So this is neural plate, that is ectoderm also, but that is thickened from inside. If you see here, this is ectoderm, but that is thickened from inside in the form of neural plate. That is neural plate border, that is called a neural crest. Now convergence. Now these inside, it will become deep from inside, like we have seen primitive streak. Now it will become deep from inside, and it bends dorsally into two ends, eventually join the neural plate borders, and that is referred to neural crest now, because that is joint here. When it is joint, that is called neural crest. So that two folds, if we see here in the structure B, these are the mesoderm, notochord, and then we can see these are going to be protruding up, and then they are going to be more and more invading, and then at last they are here it is going to be joined together and forming the neural groove. Later on this will form neural tube. So here we can see neural tube is formed by these cells, and this ectoderm is lying down, lying on up like this. And neural crest cells form components of peripheral nervous system and sensory neurons, and are all peripheral nervous system, somatic, what we can sympathetic and parasympathetic nervous system. So the closure of neural tube disconnects the neural crest, and these cells just come up on that like this. These later on, uh, just these neural crest cells, they form somatic and sympathetic and parasympathetic nervous system. So neural crest cells differentiate to form most peripheral nervous system, like the nerves are derived from them. So this notochord, that is mesodermal derived, that is the stimulant for this neurulation. The epidermis, that is just lying up on this neural tube, and these mesoderm derived somites, later on they just change into the muscles and bones, ribs, etc. And notochord degenerates and only persists as the nucleus pulposus in the intervertebral discs. Other mesodermal cells differentiate into somites and precursors of axial skeletons. Axial skeleton, uh, like we are having like a formation of the skeleton, skeletal muscles, and our some parts of like ribs and other bones. Okay, before just saying you all the best and thank you, you have to see some a video regarding neurulation. So if we see here the neurulation, and neurulation is the process in which the neural plate, a flat structure of ectoderm tissues, a vertebrate embryos folds to form neural tube, hollow tube that give rise to brain and spinal cord. So like that give rise to the nervous system, and that happens early in embryonic between days 18 to 28. Now you can see the overall process here, like that is an animation. You can see this one, all the steps. Now we'll see step by step the process of this. Like this is ectoderm, mesoderm, and endoderm. These three germinal layers which are formed during gastrulation. This is notochord, which is mesodermal derived, which is derived from mesoderm. This is a neural plate, and which later on the cells next to the notochord become wedge shaped and create a groove here, and neural plates start to move from the sides, and that forms the neural folds from here, and a neural groove from inside. Now let's keep moving closer and closer. The neural folds are just moving towards one another, and so much closer that at last two more hinge points are there. They are called, now they are neural folds touch with one another and merge. That forms the neural tube. The neural tube that later becomes the brain and spinal cord. Part of the neural folds that becomes the neural crest, and these neural crest cells later they form move towards the midline inside this, and will later on they form the peripheral nervous system, and sympathetic and parasympathetic nervous system, all the nerves and neurons are derived from that outside of the brain and spinal cord, which exist on both of the sides. Now by this process neurulation is completed. So dear students, that's it for today. I hope you understood all the early embryonic development process. Inshallah, we will see you in the next lecture. I wish you all the best. Allah Hafiz.